Hi, I'm Andreas Hummer, Marketing Director for Proteomics and Metabolomics.
Hi, I'm Bhavin Patel. I'm a staff scientist in a mass spec reagents group.
Today we are presenting a webinar entitled Investigating the AKT-mTOR Pathway with Targeted Mass Spectrometry Applications. Proteomics has long taken a backseat to genomics, primarily hampered by the greater technical challenges of analyzing proteins rather than DNA. However, understanding complexity of biology and how gene networks drive biology requires proteomic analysis. A major focus of the scientific community is the understanding of the relevant protein targets in biological or disease states. Those observations are enabled by platforms that allow us to identify and quantify multiple protein targets. The analytical challenges for protein measurements are often confounded due to the complexity of the proteomic samples, indicating the need for improved sample preparation strategies. The particular sample preparation strategies that we want to talk about today are improved methodologies that are designed to probe and quantify low abundant proteins. Modern proteomics technologies have created a new and much more comprehensive view of cellular proteins and usually provide a very large list of key regulatory proteins and pathways. For the detailed understanding of these cellular pathways, those proteins have to be targeted and verified in the functional context. As you can see here in this picture, this requires techniques that provide accurate and reproducible detection and the quantitation of the protein targets against a number of proteins in a very complex background. So why are proteomic samples so complex? We all know that in humans there are about 20,000 genes, which give rise to about 100,000 transcripts that are then translated into proteins. A lot of the proteins are regulated by PTMs, which will give rise to the complexity of the proteome, with about 1 million proteins estimated, or 1 million proteoforms. So going back to our challenge for measuring pathway proteins, how do biologists currently measure proteins in signaling? Researchers today need to consider many targets and many conditions if they want to study the entire pathway set of proteins. As this particular picture showed, it is an overwhelming amount of work, but it results in a semi-quantitative measurement. Researchers have been using Western blotting techniques and shell techniques as they are helpful for monitoring very discrete changes in the proteome, but it is a lot more challenging to observe small changes related with post-translational modifications that may be more subtle or are substoichiometric. So analytically, the variability of the reagents, particularly the ones that are raised against post-translational modifications or modified proteins, play a very big role in the confidence of the results at the end. So current best practices for most biologists is that they use at least 2 different methods for protein quantifications, which are shown in the next slides. Currently, biologists use Western blotting techniques, which is the most accessible method, but is largely semi-quantitative and can take between 5 hours and a day. Often, ELISA and the Luminix platforms are used, whereas the first is singleplex and the latter is multiplexed, which allow people to do many samples per day, but is limited by the number of targets that are available, particularly in a multiplexed format. More recently, the use of targeted mass spectrometry has been used to investigate protein and protein quantifications. Mass spectrometry provides multiplexing capabilities, is amenable to quantitative and very selective analysis. In addition to that, it can actually identify unexpected post-translational modifications and detect isoforms, sometimes also referred as proteoforms, which are typically very hard to detect by traditional methods. So comparing those 4 methods, it appears that mass spectrometry-based techniques have a number of advantages that would be well suited for the cellular pathway analysis. Because mass spec is an unbiased detector, particular strengths include that it allows you to detect proteins that cannot be detected with other techniques, as we mentioned before, something like most translational modifications. It's naturally multiplexed. It has a very big benefit when it comes to specificity, as mass spectrometry is very specific in detecting proteins or proteoforms. And most importantly, it can be used together or in combination with antibody-based methods to improve the dynamic range of measurement as a mass spec detector. So as we discussed, mass spectrometry is an unbiased detector with many benefits, one of which is the ability to observe a wide range of targets. In fact, so many targets that many researchers can feel overwhelmed by the amount of information they obtain from mass spectrometry. But we also need to consider the particular constraints of our experiments. While mass spectrometry can observe a wide range of protein abundance, some of the natural protein abundance of biological targets is much lower than what can be detected by the mass spectrometry analyzer. Like any other detection technology, mass spec has sensitivity limitations. In particular, mass spectrometry analyzers are introduced as ions. Ion suppression effects can sometimes affect the measurement. So how do we conduct a successful mass spectrometry experiment where we utilize the power of mass spectrometry for identification and quantification? We take two steps. We reduce the sample complexity and make it more accessible and easier to quantify for the mass spectrometer. We're using targeted enrichment strategy, which allows us to carry the biological context throughout the workflow. The most accurate quantification by mass spectrometer requires the addition of a stable, isotopically labeled internal standard, which can be used to generate standard curves of known concentration ranges and compare multiple samples relative within the same LC-MS run. So how does mass spec-based workflow compare to traditional Western blot workflow? We can see here in the slides that they are very similar. Described in the picture is the Western blot, where we start with sample preparation and cell lysate, followed by a separation step, transfer of the proteins to a Western blot membrane, and detection by a detector. Very similar, in the bottom panel, we have a separation device that's separating the peptides of interest introduced to the mass spectrometer, followed by the detection and analysis in software. However, because mass spectrometry usually is more sensitive and efficient in detecting peptides, an additional sample preparation step has to be undertaken to achieve mass spectrometry detection. So in addition to cell lysate, we also reduce those proteins, alkylate them, digest them, and make them ready for mass spectrometry detection. So there's a number of the mass spectrometry detectors that can be used to quantitate complex protein samples. There are two major types of mass spectrometry analyzers. On the top, you see a typical triple quadrupole mass spectrometer, which consists of a first stage, which typically selects the peptide of interest, followed by a stage of fragmentation, where the peptide is fragmented into a fingerprint for the peptide, and then a third stage of mass spectrometry, where the actual fragments are being detected. And as the peak eludes through the separation in time, it actually is recorded, and the area underneath the peak is then used to quantitate the particular peptide. We also have a second choice of mass spectrometry analyzer that is been very suitable for very complex samples and actually adds more specificity to the analysis. Similar to the triple quadrupole mass spectrometer on the top, the Orbitrap mass spectrometer on the bottom is using a separation step, peptide selection step in the first stage, a second stage where this peptide is fragmented to generate the fingerprint of the peptide, as well as a third stage where the actual peptide fragments are being detected, in this particular case with an Orbitrap mass spectrometer. The data are then presented as a spectrum and followed by the elution profile, which can then be used for analysis of measurement of the quantitative amount of peptides in the sample. So to summarize, what we have so far discussed is that we face a particularly tough challenge in proteomics and biological research focused on pathway analysis, as we're trying to measure many proteins belonging to this particular pathway that may actually be present in the sample at very low abundance. For a particularly successful experiment, we have to combine advanced and improved sample preparation strategies with the advantages of modern protein quantitation techniques such as mass spectrometry to achieve the goal of complete pathway analysis. So with that, I'll pass the presentation to my colleague, Bhavin Patel, who will be talking about sample preparation strategies.
Thank you, Andreas. We will talk about the sample prep improvements for the targeted mass spec applications. The first thing we want to talk about is target enrichment. Now, why do we need enrichment? This is a famous picture of iceberg where you can only see the tip on the surface. And the challenge of proteomics is that most only see what's above the surface. And most want to know what's below the surface as well. The reason of this challenge in proteomics is biological samples, such as serum and cells, have a very wide dynamic range. We are talking about 10 to 12 orders of magnitude dynamic range of relative protein abundance, making it nearly impossible to identify low abundant proteins over abundant ones. And of course, we make it even more difficult in proteomics by using the proteolytic enzymes to digest these thousands of proteins into millions of peptides. So we need additional measures in order to detect these low abundant proteins and PTMs. And enrichment provides that. So in this particular example, enrichment enables the better detection by mass spec. And here, what you are seeing is 12 relatively low abundant targets from AKT-mTOR pathway. If we look at those 12 target signaling pathway targets in two different cancer cell line, A549 and HCT, and do the straight digestion and run them on a mass spec, as you can see, a lot of these targets, you cannot detect them in a mass spec. But after enrichment with immunoprecipitation, you are able to detect and quantify each one of these targets. A lot of times, when you see the target, even without enrichment, you are only identifying about 2 peptides for that protein, which doesn't give you a very detailed characterization of the particular target that you are trying to study. So enrichment also enables that. After enrichment, as you can see here for mTOR, without enrichment, you get 2 peptides, but after enrichment, you can see 82 peptides, which is actually much better detailed characterization of the protein that enrichment by immunoprecipitation enables. And this is very crucial for mass spec identification and quantitation of very low abundant proteins, as they are all outlined here. So where does enrichment fit in the targeted proteomics workflow? As we see here, we have to do some sort of sample preparation. So we start with the biofluid or cell lysate or tissue samples, and we extract the protein, and then enrich the protein of interest using the immunoprecipitation. And with the Western blot workflow, you are basically, after enrichment, you can separate them by SPS page and do the Western blot to get the quantitation. But with the proteomics workflow, after you do the protein enrichment, you may or may not have to do the cleanup, depends on whether you have a detergent in your lysis buffer that are not compatible with the downstream digestion process. After digestion, you are generating the peptides from the IP eluate. And then there is another method that you can use to enrich at the peptide level using the anti-peptide antibody. And then you have to do some sort of cleanup and then measure the peptide concentration, normalize the sample, and run them on a LC-MS system. Now, if we look at the immunoprecipitation component at the protein level, and that's the most of my talk that I'll be showing the data about, the 2 major components of this immunoprecipitation is resin and antibody. And we have looked at a lot of different resins, and also we have a workflow that we optimize for antibody validations by IPMS. So I'll be talking in much more details about those 2 components. So if we look at the resin evaluation, these are the MSIA microcolumns. They are ready to use affinity capture devices for use in a bioanalysis of protein and peptide. And these columns are based on a proprietary monolithic chemistry that are densely coated with affinity ligands. And they provide effective and efficient affinity purification of target proteins that you are interested in. Some of the benefits of this technology is you can actually enable this with some automation. Also, since you are with this technique, you can reduce the non-specific binding, and that results in a much higher signal-to-noise ratio. Also, you get a very reproducible data as a result of very high consistency between each microcolumn and from lot to lot batches. And automation enables you with the time saving as well. But if we look at what's the traditional way of doing the IP procedure using the Agarose resin, this is the most commonly used method for immunoprecipitation in a field. And basically what you are doing, first thing is you are coupling the antibody to the beads, in this case is Agarose resin, and then you bring in your cell lysate or protein mixture, where a couple antibody with the beads is going to bind to the antigen that is raced against. And then you have to do the centrifuge them in order to remove anything that doesn't bound to antibody coupled to the beads. And that way you can get rid of any non-specific bound proteins. And then you can elute the antigen of the antibody by centrifugation and analyze them by IP to Western blot. So this is how the typically traditional IP procedure has been done using the Agarose resin. What we wanted to do is we wanted to ask questions that there are a lot of different IP methods available and there are a lot of different base beads available to do the immunoprecipitation. And we wanted to evaluate all the chemistry, all the bead support, and both direct and indirect IP method. So what we did here is we took antibody against EGFR and looked at all different kinds of base support. Here you're looking at polyacrylamide, agarose, or magnetic bead support. And we also looked at different chemistries. So if with the direct IP method, we can do the hydrazide chemistry, aldehyde, NHS ester, or epoxy. And we pulled down this EGFR target from A431 cell lysate. And as you can see here, that each method was able to pull down EGFR target, and you will be able to see the band on a Western blot. But relative recovery of the EGFR was different with the different methods. As you can see here, EGFR got a better recovery of EGFR using the magnetic beads that are streptavidin coated, as well as agarose bead with the aldehyde chemistry, and epoxy bead that are magnetic beads. One thing also we'll see with the direct IP method, you don't get too much antibody leaching. With the indirect IP method, you do see heavy chain antibody leached into the eluate. Now, as we know that agarose bead has a much higher binding capacity, and magnetic beads are kind of getting up to that level of binding capacity, but they are not there yet. But agarose beads are not automation compatible because it requires a lot of centrifugations with that particular protocol. Whereas the magnetic beads, you can actually, it's very easy to use, and you can get very low non-specific binding, and they are automation friendly. So for the same experimental design, if we look at the IPMS data for both direct and indirect IP method with different chemistry, what you're seeing here is the way we assess the mass spec data is we look at the EGFR sequence coverage that's shown here. Our success criteria was to have number of proteins identified less than 60 on this particular column to assess the background, and EGFR % sequence coverage to assess the recovery of EGFR is in this column. As you can see, none of this rabbit IgG was our negative control. As we saw from Western blot, it correlated very well. We can see the EGFR was enriched using all the chemistry, but % sequence coverage was higher for hydrazide, aldehyde, epoxy, and streptavidin magnetic beads. But if you look at the background, if you assess the background, we do get very low background with the epoxy magnetic beads and streptavidin magnetic beads. So agarose beads tends to give you a little bit higher background compared to the magnetic beads. So we decided to use the streptavidin magnetic bead version in order to optimize the workflow further. And here is our optimized workflow where we actually optimize for both magnetic beads coated with protein AG and streptavidin magnetic beads. Now, what you're seeing here in this particular method, which is different than direct IP method, is you incubate your antigen samples with antibody. Typically, we do it overnight. That allows ample time to have the low abundant targets bind to the antibody based on the antibody's affinity. And then at 4 degrees centigrade, and then we bring in the beads, magnetic beads, for 1 hour at room temperature. And once you have that antigen-antibody complex bound to the beads, we optimize the mass spec compatible wash buffers in order to make it compatible with the downstream mass spec sample prep. And with the both method, we do see antibody leaching in your IP eluate. But since we are doing the LC separation before we do the mass spec detections, a lot of these antibody fragments after digestion doesn't overwhelm the overall signal. Now, if we compare the characteristics of this protein AG IP versus streptavidin IP, what we find that protein AG magnetic beads are really good for screening antibodies because you can use any formulations with any carrier protein in order to compare multiple antibodies at the same time. Where streptavidin IP will require you to bioacrylate the antibody. So our recommended protocol is to screen multiple antibody using AG, and the best antibody you find then order them in a carrier-free format, and revalidate with the streptavidin magnetic beads. And overall, you get a very good success rate going from AG to streptavidin. Now, certain sample types are not compatible with the protein AG IP, such as biological fluids or very high vascular tissues. With the streptavidin IP method, you can actually use any kind of tissue samples or cell samples or biological fluids. So after we optimize this IPMS workflow, we have optimized method. The second major component within this workflow is antibody validation using this approach. Typically, antibodies are validated for IP, and then with the IP to Western blot, we wanted to see how we can validate this antibody using this IPMS approach. So why do we need to validate the antibody? As we know that antibodies are the workhorses of biological research. Globally, about $1.6 billion are spent on a protein binding reagents, and half of it, about 800 million, were spent for the use of poorly characterized and ill-defined antibodies. Not only that waste money and time, potentially also results in a not so reproducible result, and that leads into incorrect conclusions. So a lot of funding agencies and major journals will soon require new antibody details for validation data. So this is a recent paper that was published in a Nature Method from the International Working Group for Antibody Validation. This is the first effort where they have the recommendations are to use 5 different methods that are outlined here: genetic, orthogonality, correlation, and tagging, along with IPMS, in order to validate all different kinds of antibody. Now, IPMS is the only method that not only identifies the target, but also will look at the off-targets, interacting proteins, as well as the post-translational modifications. So how do we validate the antibody by IPMS approach? So this is the overall workflow that we use for IPMS antibody validation. We select targets and choose all the antibodies that are available for that target. And then from the target, we select cell models. And I will talk a little bit more details about how we select the cell models. Once we know which cell models matches with the target, we grow those cells and make lysate. And then we generate the deproteome of each cell line. And the idea behind doing the deproteome of each cell line, that gives you relative protein abundance of each target across different cell lines. And then we choose the cell line, which expresses the target at relatively low to medium abundance, because you really want to see the antibody's affinity for the target enrichment. And then we perform IP procedure using the protein AG magnetic beads without Kingfisher automation. And since these are magnetic beads, you can actually automate the whole process. After you get the IP evaluate, you subjected them to mass spec sample prep using the in-solution digestion method, and then you prepare the samples and run on LC-MS system. And then once you get the data, you can do the bioinformatics analysis in order to see whether you have enriched your target, as well as do you see any interacting proteins, and get the assessment of the background proteins as well. So in the first step, in order to select cell lines for this antibody validation effort, we looked at RNA expression data in NCI60 cell line and compared that against all the targets that we were interested in, and then selected about 12 cell lines that we can actually look at the deproteome data. And here is a representation of 5 least similar cell lines. As you can see that a lot of the targets are very uniquely expressed in individual cell lines. So combining this RNA and protein expression data, we were able to select the cell line for each target. So here we are looking at the results of all antibody against TP53 target. Here we selected 10 different antibodies for TP53, and we validated by IPMS method in a single run from the same cell line. Now, on the x-axis, you are seeing each antibody that are previously been website listed as IP validated, and we were able to validate each one of these antibodies. And the way we actually measure the protein abundance is by using the protein LFQ value. That gives you relative protein abundance of all the peptides that you have identified for TP53 protein, and take the sum of all those peptides and gives you a value as a protein LFQ value. So we also wanted to assess the reproducibility, whether the IPMS method, how reproducible this IPMS method is. As you can see here, all 10 antibodies had a CV of less than 20%. The second question we wanted to ask is not only you identify your target, but how you assess the protein that interacts, other proteins that interacts with the antibody. Because when you are doing a Western blot, you are using BSA or milk to really block everything else that's around there. Where the mass spec, you are going to see everything that's present after you perform the IP using a specific antibody. So the way we calculate a fold enrichment of a given target using this IP-MS approach is we calculate the target protein abundance in IP and divide that by total protein abundance in IP. And then take the ratio of that over target protein abundance in a whole cell lysate and divide that by total protein abundance in a whole cell lysate. So in a previous example, we looked at TP53. We have the LFQ value here, and then we look at the total protein abundance in IP, and then TP53 abundance in a whole cell lysate, because we know from the deep proteome data that what is the relative abundance of the TP53 in that lysate, and then take the total protein that was abundance in a whole cell lysate. So in this particular example here, we wanted to show what is the better way to visualize this data, because you got a long list of all the proteins you identified, but we need to further process the data in a way that we can better visualize them and calculate the fold enrichment. So in this example, what you're seeing is antibody against CDH1 was plotted against isotype matched negative control antibody using the scatter plot. On the x-axis, these are all the proteins that were only seen in a negative control antibody, and on a y-axis, all the proteins that are only seen in an IP using the CDH1 monoclonal antibody. And here you are seeing all the common proteins between the negative control as well as your target IP. As you can see, there are a lot of abundant sticky proteins. They are still present in both negative control as well as your target-specific antibody. And once we filter those out, we can see that CDH1 has the highest protein LFQ value in IP samples that were used using the CDH1 monoclonal antibody. And from this, we can calculate how much fold enrichment putting in this formula that we can see for this antibody. So all the proteins that on a previous slide on a y-axis we see besides CDH1, once we filter by using %CV cutoff, we can actually create the bar graph where you can actually see that not only in this case antibody against CTNNB1 was identified as a target protein in a red, but we also see a lot of interacting proteins that bind to CTNNB1 were also seen in our IP data. As you can see here, we see about 10 different interacting proteins that binds to CTNNB1, and the ones that APC, we see much higher fold enrichment for that is because APC has multiple binding sites for beta-catenin. There are about 12 binding sites of beta-catenin on APC. We were able to see cadherin, APC, TCF. The only target we didn't see, which is listed here, is axin using this particular antibody. So beta-catenin antibody captures cadherin and many relevant interactors that are known part of the Wnt signaling pathway. So how do we assess the antibody's affinity by IP-MS approach? And the reason we wanted to do the affinity check is to select the best antibody that has the highest capture efficiency for the target, so we can actually develop the assays use that antibody to develop the assays for the targeted mass spec application. So in this particular example, as you can see here, there are multiple antibodies that are previously been validated, IP validated by Western blot. We were able to see CTNNB1 in most of the previously validated antibodies. But as you can see here, that relative abundance of CTNNB1 across different antibodies is vastly different. So, but you also see here there are a lot of antibodies that actually has a very high capture efficiency for CTNNB1 protein. So we typically select the ones that are actually has the highest capture efficiency. Since we use this approach using the protein AG magnetic beads, what we wanted to do next is ask questions that how we can actually select the best antibody, order them in a carrier-free format, and then do the streptavidin magnetic bead IP-MS validation. Okay, so far we have seen how we can validate the antibody using the IP-MS approach. Now we wanted to put this into proof of concept to show how we can use this optimized IP-MS workflow to investigate AKT-mTOR pathway targets. Why we want to study AKT-mTOR pathway targets? Because we know that AKT-mTOR pathway is altered in many cancer types, and it regulates a broad range of cellular processes. So we chose 11 targets based from this pathway. As you can see here, they are highlighted as asterisk stars. The reason we chose 11 from this pathway because these are the most 11 targets, and we also found the alignment with ELISA and Luminex pathway kits. So we had 11 total targets from the pathway. We also selected 11 phospho targets for each one of these targets, and they are listed here in a table that targets specific phospho sites. So after we screen multiple antibodies for each one of these targets from AKT-mTOR pathway, here are some of the results. What you are seeing here is for IP antibody in this particular column for each target. This is PTM-specific AKT and the total AKT. We were able to identify not only AKT, but AKT1, but we were able to see all three isoforms of AKT in a total AKT. In a phospho AKT and phospho IGF-1R, you only identify AKT unique peptides after IGF stimulation. And this is known that after IGF stimulation, this protein gets phosphorylated and their expression goes higher. We also were able to identify relevant phosphopeptide for phospho AKT, phospho mTOR, phospho IGF-1R, and phosphopras 40. We were also able to confirm the mTOR and interacting proteins from mTOR2 complex with this approach. And we selected the best antibody for each one of these targets to perform the multiplex IP before we do the targeted mass spec quantitation. So how's the multiplex IP to targeted mass spec quantitation workflow looks like? So in the beginning part, as we talked about, we select target, total, and phospho targets for AKT-mTOR pathway targets. And then we grow the cells, stimulate with IGF, and then we identify the best antibody using the protein AG magnetic beads, then do the LC-MS analysis. From this data, we also select the peptides that we found in the discovery data and develop the targeted mass spec assay around them. Once we have the assay for each one of these targets, we take the best antibody that we identify from protein AG and bioaccumulate them, and then perform the multiplex protein immunoprecipitation using the streptavidin-coated magnetic beads. And then after doing the mass spec sample prep, we add the internal standard and perform the targeted MS quantitation by PRM or SRM. So if we look at the little bit detail about how this overall workflow looks like, we start with multiplex IP, where we bioaccumulated antibodies for 11 total targets in one assay and 11 phospho targets in a second assay. We take about 1 microgram of each bioaccumulated antibody and add them to 500 microgram of lysate. And we perform IP using the mass spec compatible IP kits that comes with the streptavidin-coated magnetic beads. After you get the IP eluate, you do the mass spec sample prep, where you reduce alkylate and digest with trypsin, and you generate peptides. At the peptide level, you add internal standard and spike-tene the thermoscientific heavy peptide, aquagrade peptide for each target. So you know exactly how much you are adding the internal standard there. And then you perform the LC-MS system qualifications, where you calibrate the machines, perform the system suitability using the known standard, and re-verify that your LC-MS system is ready before you actually run any of the calibration curves or real samples. And we typically do 9-point calibration curves that covers about 3 to 4 orders of magnitude. The lowest point we go is about 0.08 femtomoles on column. And then we do the data acquisition using the PRM method on a Q-Exactive HF and SRM on a TSQ Vantage triple quadruple mass spectrometer. And once we acquire the PRM and SRM data, we perform the analysis using the Skyline software. So as Andreas mentioned that, in order to study a lot of these low abundant targets, you have to do multiple Western blots for both total and phosphorylated forms for different conditions. All the targets in our assay for 11 phospho targets in a phospho assay and 11 total targets in a total assay, you can get quantitation of each one of these targets by just running each total and phospho assay. And here is an example of the discovery mass spec data where you are looking at how many number of unique peptides you can see for each one of these targets. As you can see for phospho assay, we confirm that AKT1 and AKT2 you only identify after IGF stimulation. IGF-1R, we also see upregulation after IGF stimulation. And we were able to see all 11 targets from this pathway in a phospho assay. In a total assay, not only you identify all the targets, but you can also see that after activation with IGF, you also identify PI3 kinase, all subunits, catalytical and the regulatory subunits. And that only happens after IGF stimulation where IRS1 binds to PI3 kinase. So that also confirms the biological model that's been used among researchers for IGF stimulation. This particular method identifies all 11 proteins using the multiplex assays. And next thing we wanted to do is we wanted to ask questions that how quantitation of mass spec assays correlates with existing technology. So we took both 11 target phospho assay and 11 target total assay and validated the mass spec assays against Western blot, ELISA kits, and there are two commercially available Luminex assay kits for AKT-mTOR pathway targets. There is one 7-plex phospho and total kit from Thermo and 11-plex phospho and total kits from Millipore. So if we look at some of the results for each one of this technology, in this particular slide, we are looking at total P70S6K. For each technique, the measurement unit is going to be different because they are each using a different detector. For mass spec assays is concentration in femtomoles per IP. ELISA is giving you an absorbance. Luminex will give you in a nanogram per ml concentration. And with the Western blot, you get the volume intensity. And we looked at 3 different cell lines plus or minus IGF. So the light color is minus IGF, and the one with the blue bar is after IGF stimulation. And AQ stands for absolute quantitation, and RQ is relative quantitation. So only Luminex and mass spec, we were able to get absolute quantitation because we can run the standard curves for these two techniques, where Western blot and ELISA, we get only relative quantitation. So for this total P70S6K target, if we look at how they correlate with each other, as you can see here, we get a very high correlation. And also we had multiple replicates for each samples with each technique, so we can even look at the reproducibility for each sample set. So overall, we get very high correlation across all techniques for the total P70S6K. If we look at the second target, phospho-AKT1, you can somewhat get, overall, you get stimulation of higher expression of AKT1 after IGF stimulation. So each technique we're able to show that, re-verify the model. But you can also see that if you look at the relative abundance of each upregulation of AKT1, each technique was giving a little bit different results. So like Luminex and ELISA correlates very well. Western blot and mass spec assays were giving you conflicting results. The mass spec, if we look at here, mass spec is the only technique that can quantify different isoforms of AKT. So if you look at the previous slide or quantitation of AKT1, with the mass spec assays, we can actually quantify AKT2 because we are using the unique peptide that are for AKT1 and 2, so we can get the absolute quantitation for each isoforms. Again, if you look at the AKT2, we get like moderate correlation. Luminex and ELISA correlates very well, whereas Western blot and mass spec assays looks very similar here. But in a previous slide, we see a little bit different results. And again, these are each technique is using different antibodies, and a lot of antibodies on a Western blot, you may be seeing the total AKT and not the AKT1 and AKT2 distinction, where mass spec assay can give you absolute quantitation of each isoforms for AKT. Now, another target, phospho IGF-1R, we see really poor correlation across all the techniques. Like Luminex and ELISA correlate a little bit better, whereas mass spec assays had a very different relative expression compared to Luminex and ELISA. Western blot, we didn't see very good results. We were seeing very high variability between replicates, and also we didn't see any upregulation as we saw with other techniques after IGF stimulation across all three cell lines. So if we look at the results comparison of all techniques for both total and phospho assays, the way we are correlating them is for the total assay across 4 techniques. And if we get a good correlation, we give 3 score. If there is a 1 out of 4 technique doesn't agree, if all 4 agrees, then we give a 4. So as you can see here, with the total assay, overall we get a very good correlation. If we ask questions, which technology disagrees the most from this, if we put it on a chart, ELISA and Western blot disagree the most frequently with other techniques. And also the agreement is generally lower for a lot of phospho targets compared to the same total target for the AKT-mTOR pathway. So in a summary, IPMS verifies the antibody performance and assesses the interactions and off-targets. Validating antibodies with this optimized IPMS workflow enables you to get the enrichment information, also gives you selectivity and flexibility to better interrogate the biology. And experiments with the 11 total and 11 phospho AKT-mTOR pathway targets across 3 different cell lines shows very comparable results for IPMS and Luminex techniques, where the Western blot and ELISA shows overall low correlation. IPMS with the multiplexing capability enables a new experimental design and also give you biological insight in a much greater depth. So thank you for attending the seminar. To find more information, please visit thermofisher.com/protein purification tool.
Thank you for your presentation, Bhavin and Andreas. It is now time for the Q&A. This is your opportunity to ask questions not only to the speakers, but also anything that you have in terms of protein purification questions. To do so, all you need to do is press the Ask a Question button on your console, type in your question, and then press Send. Let's have a look at our first question, and it asks, what method do you use for cleanup after IP? Is that something you can answer, Bhavin?
Yes. So the IP method that we have optimized uses mass spec compatible wash buffer and elution buffer. So it's actually compatible with the in-solution digestions after IP. However, it depends on what you use for in-solution digestions. We are using 6-molar urea, which you have to do the cleanup afterward. And typically we do online C18 trap cleanup, but you can actually use also offline zip tip or stage tip to clean up the peptide before you run on a mass spec.
Thanks, Bhavin. And our next question asks, what's the sensitivity and dynamic range of targeted MS assays for AKT-mTOR pathway targets? Is that something that you can answer, Bhavin, or is that more for Andreas?
Yes, for Andreas.
So what we typically see in our assay is on-column sensitivity of about 80 attomoles to about 500 femtomoles for those AKT targets in the pathway using mass spectrometry. In this case, it was an Orbitrap mass spectrometer.
Excellent, thanks, Andreas. And our next question asks, why do you think the Western blot and ELISA results for many targets disagree compared with the Luminex and Multiplex IP targeted MS assays? Is that something you can answer, Bhavin?
Yes. So we know that we use different antibodies for this target for different technologies. So the antibody that was used for Western blot and ELISA is not the same antibody that we use for Luminex and IP to mass spec assays. So the differences in antibody specificity for the target accounts for a lot of times we say this antibody is validated for Western blot, but that may or may not work with the ELISA. So a lot of these targets, the differences we see are due to the different antibody used for different techniques. The IP-MS is the only technique that actually gives you specificity because you can actually identify the target in a mass spectrometer.
Thanks, Bhavin. And our next question asks, which antibody did you use for the IPEGFR? Bhavin, is that something you can answer?
Sure. We used Thermo Fisher Scientific rabbit polyclonal antibody. The catalog number for that is PA11110.
Excellent. Thanks for that. And our next question asks, do you see different recovery for single-plex IP versus multiplex IPs? And what is the upper limit for multiplexing? Is that something that Andreas can answer, or is that more for Bhavin?
I can take on that one. So what we do typically is we validate the antibodies using the single-plex IP and protein AG magnetic beads. And we start with 3 microgram of antibody per IP. But when we do the Spectrometry magnetic bead validations, we check individually 1 microgram and 3 microgram of each antibody and make sure that whether for each target is different based on the affinity of the antibody for the target. Most of the target, 1 microgram of antibody is all you need. But few targets that you may have to go 3 micrograms of antibody because the antibody has a lower affinity for that particular target, and you want to make sure that you capture 100% of the targets from your native samples. So we do see the differences, but we validate them and make sure that we have the maximum amount of antibody that we are using in a multiplex format. And the highest we have done multiplexing so far is the 12 targets for the AKT-mTOR pathway. So that comes out to about like 15 microgram of antibody in a single assay.
Excellent. Thanks, Bhavin. And our next question asks, which method is more sensitive, ELISA or IP-MS?
I can answer this question. So what we found with the targets that we had for AKT-mTOR pathway, ELISA depends on what kit you use. You may have to start with like 50 microgram of starting material all the way up to like 400 microgram of starting material. And it actually highly dependent on individual targets and the kits that are commercially available. So there is not a fixed number that you will find. ELISA overall is more sensitive for some target, but for other targets we found they are equal with IP-MS. So IP-MS assays, we typically do it at 500 microgram of starting material.
Thanks, Bhavin. And our next question asks, can you talk a little bit about the cost of each technique and also which one is the most used technique as well? Is that something you can also answer, Bhavin? Yes.
Yeah, I can take on that one. So if you look at which technique is the most commonly used technique is Western blot. Western blot is heavily been used over the last 3-4 decades for validation of any protein targets. And the cost for Western blot will come out about like $2 to $3 per sample. Now, if you look at the ELISA and Luminex, the cost, I would say per sample, per target. So because with the Luminex, you can multiplex 10, 11 targets in a single run. So if you look at one sample and one target, the Luminex is about $4. And the ELISA is coming out, because you can only do single plex with ELISA, it's coming out about, I would say, $5 to $6. With the mass spec assays, it kind of varies. It's a much broader range, but we are still kind of working out those stuff. So.
Excellent. Thanks, Bhavin. And our next question asks, can you elaborate on why protein AG beads are not suitable for immunoenrichment of target proteins from biological fluids?
Because biological fluids has a lot of immunoglobulins as a part of the matrix, and we know that protein A and G will bind to a lot of those immunoglobulins, so it will not have enough binding capacity for the antibody that you are using and for the target.
Excellent. Thanks, Bhavin. And our next question asks, do you see interacting proteins in these IP experiments?
Yes, we do see interacting proteins in IP experiments. However, based on the wash buffer that you use, a lot of times interacting proteins you see in IP MS experiments, and a lot of times they wash out because based on the stringency of the wash buffer that you are using. The wash buffer we optimize is really not a high stringency or a very low stringency. It's kind of somewhere in the middle. So the stronger the interactor, you will be able to see it in your IPMS experiment results. But if the interactor is has a low binding affinity, they are usually get washed off during the washing step in immunoprecipitation.
Excellent. Thanks, Bhavin. And our next question asks, what type of instrument do you need for these experiments, and can a core lab help this individual set up an experiment?
So this is Andreas. I'll take that question.
Thanks.
In principle, a core lab should be able to help you if they have a functional mass spectrometer. Because the samples are enriched, as Bhavin showed, you should be able to use any mass spectrometer from a sensitivity perspective. We, of course, prefer high-resolution mass spectrometers because they will give you more specificity. Particularly if you're interested in post-translational modifications, a high-resolution detector will give you a more compelling result. If you work with a core lab, I strongly suggest that you talk to the core lab before you start the experiment to make sure that you have the correct samples prepared, and then you can schedule your analysis at the appropriate time.
Excellent. Thanks, Andreas. And our next question asks, would antibodies leached into IP eluates interfere with the downstream MS analysis? Is that something you can help out with, Bhavin?
Yes. So we spend a lot of time trying to keep the antibody bound to the beads, so you don't have any antibody leaching with this IPMS protocol. What we found that the only way you can retain the antibody onto the beads is you have to cross-link the antibody with the beads, and then you bring in your samples to have the antigen-antibody complex form. Doing so, a lot of times for the low abundant targets, your recovery is pretty much zero. You don't enrich any low abundant targets because cross-linkers somehow binds to or mask the epitope binding site of the antibody for that antigen. So the protocol we came up with is, you do the antigen-antibody complex form, and then you bring in the beads for minimum time. So you capture all the antigen-antibody complex with the minimum non-specific binding. In this particular process, we looked at different elution buffers. But if you want to get the antigen off the antibody, there is about 20% of the antibody will leach into the eluate. However, since we are doing liquid chromatography separations before we get detection in a mass spectrometer, antibody fragments after digestion doesn't interfere with the identification of the target. And we have done like a lot of IPMS samples, and we never seen that antibody actually creates any issue with the LC-MS analysis.
Excellent. Thanks, Bhavin. And our next question asks, is it possible to get peptides in the enriched IP-MS which are not present or in extremely low abundance in the input total protein fraction? Would you trust such peptides? And if you were, how would you calculate the fold enrichment for such peptides? Is that something you can help out with, Bhavin?
Yeah. So, you do at times you will see that the peptides in enriched IP-MS samples they are present at very low abundance. And a lot of times, what we typically do is we have to go back and look at the data and make sure that we confirm the MS spectra, and with the high confidence, we can include those peptides in our analysis. A lot of times, our criteria is we like to see at least 3 peptides with the high confidence from the MS spectra before we do the fold enrichment calculations. And if we don't see those peptides, we usually a lot of times exclude them. But we are looking for at least 3 unique peptides for the target that we want to calculate the fold enrichment.
Great, thanks, Bhavin. And our next question asks, is MS-based methods getting more and more popular due to its higher specificity? And the second part of the question is, what is the biggest obstacle to popularizing MS-based methods? Andreas, is that something that you can perhaps pick up on?
Yes, I'll take that. So we see that a typical core facility today processes about half of their sample that they obtain are actually IP MS samples. So the method is actually pretty popular today, and I think with the introduction of more specific enrichment antibodies, I think we're going to see an increase in this method, particularly when it comes to detection by mass spectrometers. Also, I would indicate that with the more broader accessibility of high-resolution mass spectrometers, you will see that more and more biologists will adopt this particular technique because they have a broader set of questions they ask from these particular samples, particularly when it comes to the post-translation modifications.
Excellent. Thanks, Andreas. And our next question asks, for the IP-MS identification using streptavidin magnetic beads methods, do you do digestion of the whole complex, or do you elute the proteins from beads? And if it is the latter case, what type of elution protocol do you use? Bhavin, is that something you can help with?
Sure. So we have optimized mass spec compatible IP kits, which comes with all the reagents that are necessary to perform the IP eluate, which is going to be mass spec compatible after you complete the IP. It comes with the elution buffer. So yes, we recommend that you use that elution buffer, which is mass spec compatible, to elute just the proteins of the bead. And as I mentioned earlier, there will be some antibody that will be leached in your IP eluate, but with IP LC-MS analysis, that usually never creates any problem.
Excellent. Thanks, Bhavin. And just following on from that, do you know if there are any sort of white papers that describe IP-MS methods directed to protein targets identification and sort of the methods and the reagents for each step? Is that any sort of information that you can give with that?
Yeah. So if you just Google antibody verification by mass spectrometry, you should be able to get to the white paper. It's usually the first hit. From the Thermo Fisher Scientific, and there is a title is Characterization of Antibody Specificity Using Immunoprecipitation and Mass Spectrometry.
Excellent. Thanks, Barvin. And I think we've got time for just one last question. And this one asks, could the multiplex IP targeted assays be used with tissue samples from different species?
Yes, so we recently spent some time validating this assay of tissue samples from both mouse and human. And for all the targets, we were able to validate them in both mouse tissue and the human tissue. You may have to do slightly different protocol for tissue sample prep, but it is compatible with the tissue samples.
Excellent. Thanks, Barvin. Well, I'm afraid that is all we have time for today. Remember, you can watch this webcast again at any time on demand at nature.com webcasts. I would like to thank Andreas Hummer and Barvin Patel for their presentations and for answering your questions. I would also like to thank the webcast sponsors, Thermo Fisher Scientific, and of course you, the audience, for taking the time to be with us today. Thanks for watching, and I hope you'll join us again for our next webcast.